Welcome back to Organic Chemistry Lecture. Here we are moving into Chapter 5. Chapter 5 all about stereoisomerism. So that's what we're going to talk about. Stereo meaning 3 dimensions. Isomerism, well, we're going to talk about that. Remember, isomers, that's a review. Those are different compounds that have the same formula. And there's a few different ways that you can get isomers. There are what we call constitutional isomers. That's what we talked about in Chapter 4 and a little bit earlier. That's where, again, it's an isomer. It has the same molecular formula, but you have a different type of bonding. So in other words, you maybe have in one isomer, you only have carbons that are bonded to as many as 2 carbons, whereas in another constitutional isomer, you can have a carbon that's bonded to 3 carbons. So you move around the atoms a little bit, and that accounts for the difference in the chemical reactivity of that. But there is also a scenario where you have the same formula and the same type of bonding in that molecule. But with the way that the bonds are arranged in 3-dimensional space, it can be different. So these are what we call stereoisomers. So again, not only the same molecular formula, but they have the same types of bonding arrangements, just oriented slightly differently. And that's what we're going to talk about this chapter. Obviously, there's a lot of detail here that we're going to get into, but that's the basic gist of it. Oh, hold on. There we go. So for example, we talked about a type of stereoisomer at the end of last chapter when we were talking about disubstituted cyclohexanes. So if you have, for example, 2 methyl groups on a cyclohexane, if they're on the same side of the ring, we call those cis. So they're on the same side. And if they're on opposite sides, so one is pointing up and one is pointing down, that's what we call a trans structure. So this is the first example of stereoisomerism that we have. As you can see, the bonding arrangement is the same. This carbon is bonded to a methyl group and a hydrogen. This carbon is bonded to a methyl group and a hydrogen. Same thing over here. It's just that one of them is flipped from top to bottom. So because they're on opposite sides, this becomes the trans one here. So in order for us to actually specify which compound we mean, if the substituents are on opposite sides, that's the trans one. If they're on the same side, that's cis nomenclature. So, right, so another type of isomerism that you can have, and this is the same kind of cis-trans isomerism, is that if you have an alkene, so an alkene is where you have a carbon pi bonded to another carbon, that's a carbon-carbon double bond. And here's our first example of what this would look like. We have 2-butene here. 2-butene, just like it sounds, it's going to be butane, but with a double bond, so butene, and that double bond is between the 2nd and 3rd carbon here. If the methyl groups on the 2nd and 3rd carbon are both on the same side, that is, again, cis isomerism. So we would call that cis-2-butene. Don't worry too much about that nomenclature. It's just we want to get the idea of cis versus trans so far. In the future, of course, you'll have to do this cis versus trans nomenclature when we cover alkenes in, I think, Chapter 7, so a couple chapters from now. After that, we have trans-2-butene. That would be where the methyl group on one side is pointing up, and on the other side, it's pointing down. And as you can see, like, these are clearly not the same molecule. If you try to like line them up or make them move over one another, they don't have the same arrangement in 3-dimensional space. So therefore, they are not the same molecule. And so we have to come up with a word to describe what their relationship is. And that word is stereoisomerism. We're just looking for kind of differences in the bonding arrangement. Okay, so let's go ahead and give this a shot. Here's our first example. The types of options you have for this slide is that you have 2 different pairs, right? There's a pair here, a pair here, pair here, pair here, pair here. And your options are either constitutional isomers, they could be stereoisomers, or they could be identical structures. So go ahead, give it some thought. I recommend pausing the video and going through and trying to identify each of them. And then we'll cover it when you come back. So go ahead, pause. Okay, and we are back, I hope. So real quick, we'll just kind of take a look at the first one here. When we look at this, we can see our OH group is pointed up in this cyclohexane. And in this cyclohexane, it's also pointed up. So the only difference here is basically this chair flipped into this chair. So those are the same molecule. And you know that they're the same molecule because they can just flip from one position to the other position. That's all that happened in this one. Now if we move to this molecule here, this is another butene. This is a 2-methyl-2-butene. And the question is it constitutional? Is it stereoisomer or identical structures? One of the easiest ways to answer this question is basically ask yourself, if I rotate this molecule, is it going to overlap exactly onto the other molecule? So just like in your head, or you can even build like a little model, of it, each of these, and then try to flip it and see what happens. And sure enough, in this case, if you just flip this molecule, basically 180 degrees, so you just flip it right side up, this methyl group is pointing up, and then on this side, you still have one methyl group up, one methyl group down. That's the exact same as this one here. So these are the same molecule here. So this we call identical structures. Okay, so moving on. Next up, we have these dicyclohexanes. Here we have one hydroxyl group pointed up, and here this hydroxyl group also pointed up in the equatorial position on its neighboring. And then over here we have one hydroxyl group pointed up, but the neighboring hydroxyl group is actually pointed down. So we would say that this is the cis diol, the cis version of this compound. We won't have to name it for a while, but we'll just call this the cis compound. And then this, because they're pointed in opposite directions, one's pointing up, one's pointing down, that's the trans version of that compound. Next up, we have, again, an alkene here. And the way I like to do this is, if you ever have this problem, what you can do is just identify where your substituent is on either side. So here's our double bond. We have a methyl group on this side and an ethyl group on this side. If I were to draw a line from one methyl group to the ethyl group on the other side, it crosses the double bond. So that tells me they're on opposite sides of the double bond. So this is clearly the trans structure. Here we have a methyl on one side and an ethyl on the other side. We draw a line. We don't cross the double bond. They're clearly on the same side of this pi bond. So this is the cis version of this compound. Lastly, we have this here. Now, I'm not going to do it, but you can kind of add up all the atoms and you end up with the same molecular formula. I think it ends up being 1, 2, 3, 4. So C4, H8O, and same thing over here. And so they have the same molecular formula, but clearly there's no way to get from this structure to this structure. They're not just like different arrangements. They're totally different bonding arrangements. So this is a constitutional isomer over here. So let's go ahead and clear that off. Double check our answers here. There we go. Okay, so go ahead, write that down, make sure you're on the right page, and let's keep going. Okay, so go ahead, let's try to identify these as cis, trans, or neither. We're gonna go through this a little bit more quickly because we kind of showed that on the previous slide how to do that. So go ahead, pause the video, give us a cis, trans, or neither for each of these. And let's go ahead and take a look at the answers. So the first one is cis, they're on the same side. Second up, trans, these are on opposite sides. We kind of went through this on the previous slide actually. Here, these are cis because they're both pointed up on the cyclohexane. This one's neither, because remember, basically there's no way that you can flip this, and neither of these really take priority over the other one. So there's neither cis nor trans isomerism here, because if you were to flip these substituents, nothing would change. And then down here, this is trans, because they're on opposite sides of the double bond. Maybe this is a good time to actually go over this a little bit better. I don't feel like I explained it very well, so let's take a look at this structure right here. So I mentioned already that there's neither cis nor trans isomerism here. So I just want to say here, the way that you can figure that out is, just imagine if you were naming this compound. The naming convention is going to be the same. Identify the longest chain, and you could go 1, 2, 3, 4, or just as easily, you could go 1, 2, 3, 4, and both of those would end up being the same. those are both the longest chain. And so here, in order for us to have cis-trans isomerism, there would have to be a difference if we flipped either side here. If we flip these two methyl groups, nothing changes in the molecule. And even if we flip this methyl group with the hydrogen that's up here, all that we have to do is rotate the molecule, and it ends up exactly overlaying the other molecule, telling us it's the same molecule. Okay, I hope that makes sense. Make sure to ask questions, and we're going to do a bunch of practice problems like this in the lecture assignment for this as well, or the lecture practice. Okay, so we've already mentioned, we've talked about what stereoisomers are. We mentioned cis-trans isomerism, but that's only just the first type, and it's kind of the most obvious type. We're going to get into a type that's a little bit more difficult now. So, okay, so the first thing that we have to be able to do in order to identify the next type of stereoisomerism, we want to be able to identify molecules that are chiral. Chiral comes from the Greek word for hand, kairos, and the idea here is basically like a chiral object is something that's asymmetric. So like your hand is asymmetric, right? You have your thumb on one side and your pinky on the other. And you have two hands, right? You have your left hand and your right hand. And no matter how you do it, you can't get them to overlap on top of one another. They're not identical. Like you've probably known this in your entire life, but your hands are not identical. You have your left hand and your right hand. No matter what you do, you can't get them to flip. If you flip one, all of a sudden now they're opposite oriented, so they're clearly not the same. And so that brings us, that's why we use chiral to describe these compounds. These chiral objects are asymmetric, and what that really means is that it's not the same as its mirror image. So like you take a mirror image of something and you cannot superimpose them on top of one another. Okay, so this is the key kind of word that we're going to use a lot in this chapter, which is non-superimposable mirror image. That sounds like a mouthful, but it's actually just really an easy way for us to talk about these chiral centers. Non-superimposable is exactly what it means. To superimpose something means you just are able to put something exactly on top of something else. So like if you were to superimpose, here we go. So I've got two little atoms here. What we're gonna do is we're just gonna superimpose right on top of one another. That means in our heads, what we're doing is we're taking them and we're merging them together. And if they are exactly the same, that means they're going to match up exactly the same in 3 dimensions. The key is some of these are going to be non-superimposable. So non-superimposable, that's the word we want to use here. That's just like here, we have a green and a white here. When we put them on top of each other, they're not the same. So we superimpose them in our head, they're not the same. That's non-superimposable. Now the second key is mirror image. And mirror image, we're going to show a few examples of that to kind of show what that is. So yeah, so that's the ultimate trick here for organic chemistry and stereoisomerism of this type. You're just going to see whether or not two objects are superimposable. Okay, so yeah, so you may ask yourself, like, what's the point? Like, why do we even care about this? Well, it turns out that most of the biomolecules that you have in your body are chiral. So like proteins, for example, amino acids have a particular chirality because the carbon in them, in each amino acid is chiral. And so basically all of our amino acids have to be oriented in the same direction in 3 dimensions or they don't fit. In fact, if they don't orient themselves in the correct way in 3 dimensions, they're the opposite handedness, they can be poisonous. So you have to be very careful about that. The same thing with carbohydrates. The same thing is also true with our DNA. Parts of our nucleic acid are chiral as well. And that brings up, if our body's biomolecules are chiral, that also means that the drugs that we put inside of our body may also be chiral. And if they're chiral in the wrong way, they may have very awful consequences. They may, again, be poisonous at the very worst. So yeah, so this is going to be just kind of like an example, like you want to really be using 3-dimensional models to help you through this. So I recommend get your model kits out and just kind of build the models as we go through here. Okay, so first up, go ahead, grab a carbon, make sure your carbon has 4 different bonds, and try to build, just like grab 4 random colors. Like here what I have is, I have my carbon in the middle, it's bonded to a white one, a yellow one, a red one, and a blue one, okay? And then what you wanna do is go ahead and make its mirror image. So like blue on the inside, yellow on top, red facing you, white facing me, like that. Build its mirror image. Go ahead, pause the video, build that up, and then we're going to go ahead and see, are they superimposable or not? And it's crucial, again, your carbon atom has to be bonded to 4 different colored atoms here. Okay, so now let's go ahead and try to superimpose them on one another. This is easy enough, right? It's just you're rotating things in 3 dimensions. So let's see here. So I know yellow's on top, blue has to be pointed to the right. Okay, so I'm going to flip this one like that. Then I try to superimpose it. And okay, yellow matches yellow, blue matches blue. But then, oh, over here, white is on top of red. And then back here, red is on top of white. Huh, okay, so I guess I can flip it again, right? And then, okay, great. So now we have white on top of white and red on top of red. Whew, goodness. Oh, crap. Oh my goodness, look at this. Now, but blue is on top of yellow and yellow is on top of blue. Oh, but so I can just flip it again, right? And then it's like, okay, I hope you get the idea here, basically, that no matter how you flip these, no matter how you rotate them in 3 dimensions, they're never going to be superimposable. They're never going to be the same molecule because of the orientation in 3 dimensions. And so this is the real key thing. We need to be able to identify in our molecules when we have a carbon center that's bonded to 4 different groups of atoms. Okay, so let's go ahead and take a look at some of these. So remember, carbon is tetravalent. It's going to bond to 4 different groups, and so it's going to be tetrahedral. And when it's bonded to 4 different groups of atoms, that's when we call it a chiral center. So looking here, let's see if we can recognize this here. This carbon is bonded to a chlorine, it's bonded to a methyl group, and it's bonded to an ethyl group. That's 3 different ones. We also assume it's bonded to a hydrogen. So count them up. Chlorine, methyl, ethyl, hydrogen. That's 4. Ding, 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 ding. Okay, moving on. This carbon is bonded to an ethyl group, a propyl group, a hydroxyl group or an alcohol, and also to a hydrogen. So hydrogen, ethyl, propyl, hydroxyl, ding, 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 ding. That's 4. That's a chiral carbon. Here, this carbon is bonded to a bromine, then it's bonded to the left side of this ring, and it's bonded to the right side of this ring. Now, if this ring were the same all the way around, that would be the same thing. But as we can see, that's not true. This ring is asymmetric. It's got a double bond on one side and no double bond over here. So we've got bromine, hydrogen, and then double bond side of the ring, non-double bond side of the ring. Ding, 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 ding. That's 4 different ones. That is a chiral center. So this is something that you're going to just have to practice. It also helps if you like draw in the hydrogens at this point, and you can kind of see, oh, this one has a hydrogen on it. That's going to be its 4th different group. Okay, so go ahead and ask yourself, how many chiral centers are in each of the following compounds? Give the video a pause and see when we come back. All right, so let's go ahead and take a look through here. I'm going to go ahead and change this up. Oops, draw. Okay, here we go. So we're going to look at certain carbons and we're going to ask ourselves, are they chiral or are they not? So again, we're looking for 4 different groups. So starting here, going around this ring, this is cyclohexanol, doesn't matter, but that's what it is. Each of these carbons is bonded to like CH2, 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 CH2. Those all have 2 hydrogens on them. Because they have 2 hydrogens, they can't be chiral, right? No matter what, this one's bonded to hydrogen, it's bonded to CH2 around this ring, and CH2 around this ring. That's only 3 different things. That's not chiral. So that's not what we're looking for. Obviously, we're going to evaluate this carbon here. So this carbon is bonded to an OH group, so that's 1. It's bonded to a hydrogen, that's 2. Now we have to ask ourselves, we're going around the ring here. So going around the left side, it's CH2, 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 CH2. Going around the right side, it's CH2, 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 CH2. Those are identical, believe it or not. So this carbon is only bonded to 3 different groups. Therefore, it is not chiral. So we're going to put a big old X through there. All right, moving on to the next one. So here we have the same thing, but now it's diol, so cyclohexane diol basically, and their neighboring alcohol groups. So obviously, again, this is CH2, 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 CH2. None of these can be chiral because they're all bonded to 2 hydrogens. This carbon, though, and this carbon are potential. Okay, so now let's check it out. This carbon is bonded to a hydroxyl group. It's bonded to a hydrogen. It's pointing straight down. And then it's bonded to, on this side, CH2, 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 CHOH. And on this side, CHOH, CH2, 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 CH2. That's two different things. So guess what? This is a chiral. So we're going to go ahead and check mark that one. Same thing up here, right? We know that this one's also got a hydrogen sticking out over here. So it's got an OH group, a hydrogen, that's two, easy right away. Then it's bonded to CHOH on this side and CH2 on this side. That's four different groups of atoms. That's a chiral center. So then we've got two chiral centers on this one. And this one, sorry to answer, that's a 0 right there. Okay, so now let's go ahead and take a look here. So, okay, one big clue that you're going to look for and you're going to kind of see on future tests probably is looking for wedge and dash notation. If I'm going to tell you that something is chiral, then you're obviously going to need to see the wedge and dash notation for it. Now that doesn't tell you everything about it, but it gives you a good clue that something might be up. So it's a good place to look. So first thing in, let's go ahead and draw in our hydrogens. So here we know that these two methyl groups, so just kind of like drawing it in here. If you have these two carbons that are in the same plane, oh, and let me grab one more carbon. So like evaluating, for example, this carbon here, these two carbons are in the same plane. This carbon is sticking out towards you, so it's going to go like right there. That tells you that, oh, in the back, that's where that hydrogen is. So you can just draw a dash there. Remember, these carbons are each tetrahedral, and now we're trying to draw that in the 3 dimensions. So I hope you had some good practice on that in chapter 2. If not, now's a really good time to kind of brush up on that to help you out. Oh, and then this carbon here also has a hydrogen on it. That's another one that we want to evaluate here. All right, go ahead, pause the video, come up with your answer, how many chiral centers you have, and then we'll evaluate it as when you come back. Okay, so first up, let's go start with this one. This one is bonded to a methyl group. It's bonded to an ethyl group. It's bonded to a hydrogen. And then it's got this big old thing over here. So that's like that's a butyl group of a sort. So it's, or I should say it's a disubstituted propyl group of a sort. That's definitely something that's different. So that's 4. So this one for sure, chiral. That's 1. Okay. Next up, we're looking at this carbon here. The carbon's bonded to a methyl group, a hydrogen, the isopropyl group over here, and then the sec-butyl group over here. That's 4 different things. So guess what? This one's also chiral. Now looking at the first carbon on the left, this one is bonded to a hydrogen, this big old thing over here, a methyl group and a methyl group. Wait a second. So this big old thing plus hydrogen, and then a methyl group and a methyl, that's only 3 different things. So that one is not chiral at all. So the correct answer here is you have 2. All right, so then finally we've got this one here. So go ahead, add in your hydrogens. Okay, and ask yourself chiral or no chiral for each of these. And let's go ahead and take a look. You can pause the video if you want here. All right, so we're looking again 4 different things here. So this carbon has a hydrogen bonded to it. It's got a methyl group bonded to it. It's got this weird thing bonded over here, and it's got an ethyl group. So that's 4 total things. This is a chiral carbon. So we're circling that one. This one here is bonded to a hydrogen. It's bonded to a methyl group, bonded to a sec-butyl group, and then this alkene over here. That's 4 different things. So this is also chiral. So the answer here should be 2. Let's go ahead and clear them up. There you go, not too bad, right? So that's how you kind of evaluate chiral centers for these different compounds. Make sure to get some practice in the lecture exercise down below. Okay, now there's a special class of stereoisomers that can be called enantiomers. Enantiomers are these non-superimposable mirror images that we've been talking about. So for example, if you still have your model kit around, the ones that we were playing around with earlier, the mirror images that look like this, these are non-superimposable mirror images. These are enantiomers. So like here, we take this chlorobutane, 2-chlorobutane. If you take the mirror image of it, this is going to be its enantiomer. And sure enough, if you try to imagine like flipping this one so that it matches up with this compound here, that chlorine is going to stick in to the screen, and this chlorine sticking out of the screen, you'll never be able to superimpose them. So they are different. So the easiest way to do this, if you're going to draw it, rather than like drawing a mirror and then like going through it that way, you can actually just go ahead and like invert one of the wedges or dashes. So like the easiest thing is like, okay, we've got this chlorine sticking out and it's an enantiomer, we're going to stick it in this way. Same thing here with this amine. We have this methyl group sticking out. Well, just like flip it. You know, this methyl group's now going to stick in. Here, this is isopropanol here, isopropyl alcohol, rubbing alcohol. This carbon is not chiral, right? Because it's bounded to two methyl groups. So like flipping this wedge doesn't really do anything. You can imagine taking this isopropyl alcohol that's sticking out towards you, you flip it this way, it's going to superimpose exactly over this isopropyl alcohol. So it's an identical structure. So because there's no chiral center, the mirror image is identical. Okay, so now we come to nomenclature. So this is kind of the key part of this chapter that we want to make sure we understand. So if we agree that these enantiomers or these different chiral centers are differently arranged in 3 dimensions and they have different reactivities, which they do, they then are not the same compound. You can't call them the same thing because otherwise you're going to get things mixed up. And when you get things mixed up in terms of chirality and pharmaceuticals, for example, you can turn something that's a miracle cure into a poison. So we have to designate basically a system to come up with this. And what we use is what we call the R versus S configuration. And in order to figure out what the R versus S is going to be, we use what's called the Cahn-Ingold-Prelog system to designate the substituents of each chiral center. So here what we mean is like we're looking at each of these chiral centers and we're going to go ahead and give each group that's next to it a number. Okay, so one thing to note is if you have a compound that has the R configuration at its chiral center, when you look at its enantiomer, it's going to be flipped into the S configuration. But we'll cover that here in a second. It's going to make sense in a second. So here's how you do that. So here's the list of rules, and we're going to do a bunch of examples of this. So don't worry, there's a lot of practice. Basically what we're going to do is we're going to look at the chiral carbon and look at its 4 things that it's bonded to. Each of those is going to get a number from 1 to 4. And we're going to use this Cahn-Ingold-Prelog system to give us that number 1 through 4. It's really simple. Basically, whatever has the biggest atomic number gets the 1, and the smallest atomic number gets the 4. Then what you're going to do is you're going to go ahead and look at the molecule, and you're going to arrange it in 3-dimensional space, either with your little model kit, for example, or in your head is where I recommend eventually getting to it. And then what you do is just count the group priorities 1, 2, 3. So you're going to orient it so that number 4 is facing away from you. You can't see number 4. So like for me, for example, like number 4 is almost always hydrogen if something's bonded to hydrogen, because hydrogen's atomic number 1. That's the smallest number. So we're going to orient it away from you like that. And then you're just going to go ahead and count. So if this is like chlorine, let's say, that's going to be number 1. This is oxygen, that's number 2. Or sorry, this should be nitrogen and oxygen. So 1, then comes oxygen, 2, then comes nitrogen, 3, and then 4 is facing away from you. What you're going to do is figure out, is that going clockwise or counterclockwise? So going 1, 2, 3, that is going, this is really tricky for me because I'm looking at it the opposite way of you. That is going clockwise, right? Wait. Yeah, clockwise. Sorry, it's very tricky to flip things around like that. So a clockwise is actually going to be what we call the R configuration. Counterclockwise is going to be the S configuration. Now let me give you a little hint here. So this is going to help out a little bit. So I know that's a lot to talk about without actually doing an example, but real quick, I'm going to tell you the trick for R versus S. So the way you do it is basically like if you're going clockwise, so you're going clockwise like this, right? So clockwise like R is like that. Boom, that's an R. It's like the top of the R. Counterclockwise though, going like this, that's like the top of the S. So it's like you kind of complete the letter here for the S from the top, and the clockwise is with the R as you draw it. So that's the way that I remember that, and I hope that kind of makes sense. It's a lot easier to share like on the board as we're looking at things together, but I'll give it a try on the next slide too. Okay, so for example, this chiral center is bonded to a chlorine, an oxygen, a carbon, and a hydrogen. So we have to number 1, 2, 3, and 4. Clearly it's a chiral center, it's bonded to 4 different atoms. So we know that hydrogen is going to be the last number because the higher the atomic number, the higher the priority is going to be. This is what we call the Cahn-Ingold-Prelog system. So hydrogen with atomic number 1, that's always going to be the lowest priority for us. The highest priority here is going to then be between chlorine, oxygen, and carbon, just evaluated based on its atomic number. Chlorine has the highest atomic number, then oxygen, then carbon. So this is going to be 1, 2, 3. So that's the way it goes, basically. The higher the atomic number, that gets the number 1 priority, then oxygen gets 2, carbon gets 3, hydrogen is 4. Now what we want to do is we want to rotate it so that the hydrogen faces away from us. So the way that I do this is I try to do it in my head, basically. You can just kind of like flip it in your head, and then you designate, okay, it's 1, 2, 3. So I'm looking at it from the opposite perspective, and that's going to tell me which way we're going to go. You can also use a three-dimensional model that can help you out a lot as well. So here what we do is, for example, we do have 4 facing us, but we need to go ahead and flip it back. So we're going to go ahead and rotate the whole molecule. Now that means that 4 is going to be in the back, 1 is going to be at the top facing us, and then we also flip 2 and 3 down below. So whereas 3 was on the left, 2 was on the right, now all of a sudden 2 is on the right, 3 is on the right. 3 is on the right. 2 is on the left, 3 is on the right. So now what we want to do is, again, you can use a handheld model at this point. This can be a really helpful situation. Just say, okay, so I've got chlorine, that's my number 1. Hydrogen, that's my number 4. So it's going like that. And then down here, carbon like so. And then oxygen, that should be the red one. That's right there. So now my model, if you look at it this way, that matches what you see on the screen. So then I'm going to orient it in 3 dimensions so that now the hydrogen is pointing away from me. Same thing here. You can look at it this way as well. Okay, and now we're ready to do the clockwise versus counterclockwise thing. We have to orient it so that 4 is facing away from us, so facing away from you, in other words, into the screen. And now all of a sudden we can evaluate. So we've got chlorine, oxygen, carbon. That's 1, 2, 3. So we go ahead, 1, 2, 3, do the little circle like so. And that is going to be counterclockwise. So counterclockwise, that matches S. So the way I always think about it is like, basically look at it this way. Again, the more you practice this, the better off you're going to be. So it's 1, 2, 3, like that. That's like the top half of an S. That's how I kind of like evaluate that. So now we know that the chiral center that we have has the S configuration. Okay, so now it can be tricky if we have like chiral centers that are very similar. So like earlier we had the difference between like an ethyl group and a propyl group. So in other words, here, like here we have a sec-butyl group, and over here, it's going to be like a pentyl group or a hexyl group down here. What you have to do is you have to analyze the atomic numbers 1 layer at a time. So like here, this is a CH2, this is a CH2. That's a tie. So we can't evaluate which one's 2 or 3 until we break that tie. So now we go to the next carbon over. The next carbon over, okay, now this carbon is bonded to 2 other carbons. So CH is what it looks like. And this carbon's only bonded to 1 other carbon, CHH. So this carbon, because it's bonded to more carbons at this step, that's the tiebreaker. So now this gets the 2nd priority, this chain over here, and this one gets the 3rd priority over here. So again, evaluate it 1 layer at a time. Don't look like way far away and say, oh, this one's got more carbons, that's good. Go 1 step at a time and see where the tie breaks. The closer it is, the more important it is to the molecule. And basically, it's like, again, here, the priority is based on the first point of difference. So here, this is a carbon, and this is a carbon. The 2nd layer, now all of a sudden, this is an oxygen versus now it's bonded to 3 separate carbons. So this oxygen, that's a higher atomic number. That's going to be the tie break there. Okay, go ahead, label the chirality centers in vitamin D3. Try to figure out where they are, and then we will come back and I will go ahead and go through it with you as well. So give it a shot, take a break. Okay, so I went ahead and counted up 1, 2, 3, 4, 5 chiral centers. Just briefly looking at it, again, what you're trying to find is carbons that are bonded to 4 different possibilities. This is kind of like, this probably should have been earlier when we were talking about chiral centers. I don't think it really belongs at this point in the lecture, but let's go ahead and do some R versus S next, is more important. Now remember, okay, so this is one other thing about the Cahn-Ingold-Prelog system. When you have double bonds count as 2 single bonds. So this carbon is actually bonded to 2 oxygens is the way that we think about it in terms of like tiebreakers. So this is like a double tiebreaker for this one with 2 oxygens here. So now this one is bonded to a CH3, it's bonded to a CH2 and a CH2, and this is bonded to a COOH. So that just like is going to break a few ties here and there. Okay, so now some strategies here. There's a couple different ways that you can do this. One is that we mentioned the tricky part is making sure that the lowest priority group is facing away from you. If you build a model, you'll be fine. Like you can just orient it in 3 dimensions, it's no problem. But there is another trick that works. One is that if you can just imagine flipping the groups, if you just switch 2 groups alone, and you can usually orient this such that the lowest priority group is away from you. That's going to be your goal. If you flip it that way, you can evaluate it just on paper what the chirality of that center is going to be. And if you just flip once, that flips the orientation of the chiral center. So in other words, if you were to flip groups 1 and 4 here, that takes you from the S configuration to the R configuration. That any 2 of these groups you can flip. You could have flipped 4 and 3, 2 and 1, for example, and you would get the exact same situation. Now what you can do is, you can imagine flipping once, okay, so that gives you a different orientation and moves the 4 to the back. Then you can flip again. And when you flip again, all of a sudden, now you're back at the original orientation away from you. So you use the first flip to move the lowest priority group back. Then either you evaluate at that moment and say, okay, this is the opposite orientation, so I know my real orientation is the other one, or flip again, and then you know that you're still evaluating the same orientation as what you started with. So I hope that makes sense. Again, the more practice you get with this, and we'll be doing some examples in the lecture exercises as well, the more practice you get, the better off you're going to be. So yeah, here's your kind of like CIP rules summary. Identify the 4 atoms, assign a priority to each of them. If they have the same atomic number, you're going to move away 1 layer at a time. And the first layer that is different is the tie break. Then you rotate it so that the 4th priority is away from you. And then determine the 1, 2, 3 sequence. If it's clockwise, it's R. And if it's counterclockwise, it's S. So go ahead. We've got a chiral center here. Give it a shot. I'm going to pause the recording now, pause the video. And then when you come back, we're going to go ahead and take a look at it at that point. Okay, I hope you took a chance to pause the video, and let's go ahead. So clearly the chiral center I mentioned, you want to look at the dashed and wedges first. That's a really big hint. So here we've got this NH2 going into the screen, so coming out towards me. That means the hydrogen is pointing out towards you, like that. Okay, so let's check and see. Okay, so this is a carbon bonded to a carboxylic acid on one side. This whole thing over here on this side of an amine and a hydrogen, that's going to be a chiral center. That's 4 different things. So let's go ahead and assign a priority. So the way that we can do this is, let's see here. So we've got the first layer first, right? So we've got hydrogen here. That's definitely going to be number 4. So we're going to give that a number 4. Then it's nitrogen, carbon, and carbon. Well, the biggest atom is going to win in terms of the atomic number. Nitrogen has 1 more proton than oxygen does, or than carbon does, excuse me. So that means that nitrogen is actually the winner. It's the number 1. So this is where things get tricky, right? Like you may be tempted to assign 1 over here because of these oxygens, but you're evaluating it layer by layer. And these are both carbon. So that means that they're tied to the first layer. And this is a nitrogen. So that means this is number 1. Okay, now breaking this tie should be pretty easy, right? This carbon is bonded to 1, 2, 3 oxygens according to the Cahn-Ingold-Prelog rules. This is just bonded to 1 carbon here. So this is clearly going to win the tiebreaker there. This is number 2, and this is number 3. Okay, so now when we're kind of drawing things out here, we've got 2 and 3 up like that. And then we've got 4 facing us and 1 going into the paper like that. Okay, so now this is where we have our kind of like flip rules, right? So now we can imagine, let's just go ahead and flip. Here, let me use a different color here. Let's flip 1 and 4, right? Because we want 4 to be in the back anyway. So let's go ahead and flip that. That gives us 2, 3, 1 facing out at us, and 4 going in. Okay, so now we know that this one is going to be the opposite orientation. So this is opposite. So what I do is, for me, I just stop here. I'm very lazy as far as like drawing things out goes. I stop here and I just know, oh, it's going to be the opposite of whatever this one is. So for me, what I do is I just say, oh, okay, it's 1, 2, 3. So we go around like this. And I just imagine in my head, I'm going around, around, around. Oh, that is clockwise. And that looks like the top half of an R. So this was the R. The R is the opposite. That means that the original one is S. Something like that. Now, again, you could do what the book recommends, and you just flip again. So, oh, sorry, let me go back to the green ones. Let's flip the 2 and the 3 here. So now we've got 3 over here, bonded to our chiral carbon, a 2 on the right, 1 still pointing out, and 4 going in. Okay. So now we go 1, 2, 3 like this. And so when I do this now, all of a sudden that is counterclockwise, and that is like the top half of an S. So we know that that matches up to our original molecule here, S. Okay, so yeah, so that's how you do that. Hold on. Take some time, kind of work through it. This is going to be something that we're seeing a lot this semester, so the more practice you get on it now, the better off you're going to be. Okay, all right, next up. This is almost the very end. I think there's just a couple things we want to say here. When you run into these in nomenclature, what you do is you actually put the R or S in parentheses at the beginning of the IUPAC name. So this is like parentheses R2-butanol. This is parentheses S2-butanol. And then if you have 2 chiral centers in the same one, you're going to put both of these in parentheses. And you're going to say, okay, carbon 2 is R. So I say 2R, comma, carbon 3 is S, 3S, end parentheses. And you just put that at the very beginning of the name, separated from the original name by a dash. We'll get some practice on this in a little bit here. But one thing is that we want to keep in mind that, and this is something we're going to talk about in the next set of slides here, is about optical activity. Basically, because the structures are only different in the way that your hands are different, the only differences between them is going to be basically they're going to have the same physical properties for the most part because they interact with each other in the same way. But how they interact with light is different because light travels at different speeds based on the electron density in a given location. So that changes how light can interact with it. We'll talk about that. That's what this word optical activity. And then also how they interact with other chiral compounds is going to change. And that goes back to our biochemistry. So we're going to talk more about that in the next lecture. But for now, take a breath, work on that lecture exercise, and try to figure out this S versus R thing. We'll come back in lecture 17 with a little bit more. Until then, I'll see you soon.